Hey guys, let's finish the second section of chapter 9, and then we'll have one last section at the very end. This time we talk about DNA replication. Previously we talked about DNA, its structure, what it's made out of, how it bonds. Now we're going to talk about replication. How does it copy itself? We mentioned mitosis and meiosis in previous chapters, and mitosis, we were going to make a copy of the cell. Well, to do that, remember we had S phase of interphase. That's what we're going to talk about. How does DNA copy itself? How does it make a copy for that S phase of interphase? So let's get to that. Remember, these guys are complementary strands, meaning they're going to have one nucleotide bind with the other nucleotide. A, adenine, is going to bind with thymine. A's and T's are together, cytosine and guanine. C's and G's are always together. They are complementary. Another thing to remember is that they're anti-parallel, 5' and 3', and 5' and 3', and I'm going to flip them. There we go. So these guys are going to run anti-parallel, 3 and 5, 5 and 3 for these guys. How does DNA replicate? Literally, like how does it? Does it do that? We didn't know until, again, the 1950s, with those experiments that we talked about using crystallography, about the basic structure of it. But how does it copy itself? Does it like split in half? There were 3 different theories that, we take the original one and it copies off of a whole brand new one. So everything in blue in this picture, these blue ones are new, and the gray one is the old original one. So it just copies itself totally over. We have an old one, original, and then it just makes a brand new, absolutely new one, and keeps the old one intact. That would be considered conservative. Then we have this other theory where the DNA strands, when they copy themselves, they copy in chunks, and this chunk of DNA and this chunk of DNA, like these group of genes and this group of genes and this group of genes, and those guys would be called dispersive, where we have old stuff absolutely mixed in with new stuff. Old stuff, new, old, new, that's dispersive. But it turns out that it is actually semi-conservative. And we won't go into the science behind that, or the Hershey and Chase, the scientists that did this experiment to figure out that it is semi-conservative. But I do want you to know that that is how DNA replicates. That is considered semi-conservative, because conservative would be keeping all of the old one and creating a brand new fresh one over here. This is semi-conservative. So we're going to still hold on to the old one. Here is our old one. So here's our guy here and here. And those would be our templates for creating the new strands here in blue. So semi-conservative is where it's at for DNA replication. What are the steps of DNA replication? You'll need to know these 3 basic steps and kind of what happens in here. There's a lot of detail in these 2 videos here. The first is Bowman Science, and the next one, I can't remember who the author of is that video that I found on YouTube. Is another one out there that has the whole process, excuse me, done for you in like 5 minutes. So let's talk about the 3 basic parts. So we're going to copy a DNA strand. How does that happen? We break it into 3 groups of labor. So the first one is initiation, and that's where we're going to start. DNA replication is going to start at a place called the origin. Here's our origin right here. So that's where DNA replication begins. So with the origin, we can see it's right smack here. Here's our little line where the origin is. So this is the location on the DNA where replication begins, is the origin. And so once we find the origin, then we are going to have an enzyme that's going to come along and bust up those hydrogen bonds that keeps, A's and T's together and C's and G's together. We talk about those hydrogen bonds. There were 2 and 3 of them, depending on which nitrogen spaces there were. They're going to break those bonds. So helicase comes along. Initiation is going to involve helicase. It's also going to involve there's other guys in here that's a little more complex than what I'm writing down for us. But I do want you to understand that helicase is going to come along, and it is going to unwind. Helicase unwinds the DNA. It gets in there and splits it. Look, it's like a giant wedge that comes along and it just trucks in and splits apart the DNA. It's going to break those hydrogen bonds. And now instead of one zipped up strand of DNA, this double-stranded helix, it's going to split. It starts to split because of helicase. And then there's going to be single-strand binding proteins that keep those things open so they don't stick back together, but those are not in your outline. So we're not going to worry about single-strand binding proteins. We're just going to learn about helicase. And helicase splits it apart. And that is part of initiation. Next step is going to be elongation. That's kind of the exciting phase. Elongation. And elongation is going to be where we lay down the new DNA. So we said this was semi-conservative. We're going to keep an old strand and then we're going to make new stuff that's going to be the complementary side on the other side. So with elongation, we are going to have two things that we'll need to know. Primase is an RNA primer. Primase lays down these little RNA primers. So those are going to be shown in green. So this guy, here it is. Here's a little sign for it. This guy, all of these things in green, these are RNA primers. We have to stick them down first to prime the location, hence the name, because we're going to have another guy, primase with an E. We're going to have another guy come along, and this one is DNA polymerase. I'm just going to shorten it up, DNA pol. There's DNA polymerase 1, 2, 3. There's different DNA polymerases, but I just want you to understand what DNA polymerase does. DNA pol is going to lay down the new DNA. Primers. Lays new DNA. Lays down new DNA. It's always going to lay it in a certain direction too. We are going to lay it in 5' to 3'. It's going to always run 5 to 3 every time when DNA pol is laying down new DNA. So it can only go in that direction. So DNA pol comes along, it just sticks on to those RNA primers. That's the location that it's looking for. It cannot lay down new DNA without an RNA primer. So primase lays down the primers, DNA pol comes along, latches onto that primer, and then starts trucking along like a little choo-choo train on a track, laying down new DNA in the 5' to 3' direction. Do not forget 5' to 3'. That's how DNA pol lays it down. So if you see these guys, we can see that there were green spots where the RNA primer is, and then there is the new DNA being laid down in red. And if you notice that some of these areas are going to have long stretches and some of these are going to have short stretches. So the long stretch, this guy is called the leading strand. It's the leading strand because DNA pol is going to lay down the polymerase in this one direction. Remember, it lays down in 5' to 3', which is perfect because this strand that it's laying it on, this is 3' here and 5' here. So remember we said they were anti-parallel. This is perfect. Our DNA pol is going to stick onto that primer that's stuck on at the origin. So here's your little primer stuck on right at the origin. DNA polymerase sticks onto that primer and then starts running, laying down the new DNA in the 5' to 3' direction. So this is perfect for this leading strand. On the other end, this other strand on the opposite side, here is its 5' end and here is its 3' end. So if DNA pol lay down here and went this way, it wouldn't work because that's not the way DNA pol runs. DNA pol has to run from 5' to 3'. So this is called the lagging strand. The lagging strand. Here it is here on the other side. Here it is up here. This is the lagging strand. Here is a leading strand on this side of the origin. All right, so on our lagging strand, DNA pol is going to have to do it in chunks. It's still going to lay it in 5' to 3', but it can only lay it in little pieces because the strand is backwards. So it lays it in these little pieces, and those little pieces are called Okazaki fragments. Okazaki fragments. So here's an example of one of those pieces on this lagging strand opposite of the leading strand. And those pieces are Okazaki fragments. So we're going to lay them down, lay them down, lay them down. That is called elongation. So 1 was initiation, step 2 is elongation, and step 3 is going to be ending it, stopping this process. So that is termination, termination. And termination, we are going to, DNA poll is going to run, 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 and eventually get to where it can't go anymore and it stops. And then we are going to glue our pieces together. What were our pieces called? The fragments, the Okazaki fragments. We need to glue those together, so we're going to have a seamless piece of brand new DNA laid down next to the old one. So we are going to use an enzyme, DNA ligase. Ligase acts as a glue. So ligase is going to come along, it's going to remove all the RNA primers, it's going to pull off all the green stuff. It's going to pull it off. And when it does that, it's going to stick down more DNA. So it's kind of like DNA pull a little bit, and the fact that it can remove the primers and then fill it in. But that's the job of this. It's basically the glue that's going to stick this chunk and this chunk, and we're going to seal them together with primase, glue all those fragments together so we have this beautiful new strand that lines up next to the old strand, and a beautiful new strand up here lining next to the old strand up top. So let's go through this one more time. We are going to have the DNA, and where it begins its replication process is a place called the origin, and at this origin, it's going to start to split. It is the first part of this process is initiation, and an enzyme called helicase comes along and wedges itself in here and splits open the DNA, breaks the hydrogen bonds, splits open the DNA. That is initiation. We have started the process. We have initiated the process. That's the beginning. So we started at the origin. Helicase came along, wedged itself into those hydrogen bonds, busted them open, and now DNA is split open. What do we do now? Now we need to elongate and lay down new DNA. So elongation is step number 2, and it is laying down new DNA. We can't lay down new DNA without a primer. So primase is the part that is going to lay down the RNA primers in green, and those give a place for the DNA pole to attach. DNA pole is going to lay down new DNA. DNA pole always lays down, again, 5 to 3, 5 to 3, 5 to 3, 5 to 3 every time. So remember that. Because it lays it down 5 to 3, it's going to have a leading strand and a lagging strand. And on that lagging strand, it's in chunks, it's in pieces called Okazaki fragments. Once we've laid down the DNA as much as we can, we've gotten to the end, DNA pole can't lay down any more DNA there, we are going to end it. We are going to terminate this process. Termination is going to involve another enzyme called DNA ligase. And DNA ligase acts as basically the glue. It's going to remove the RNA primers, and it's going to lay down DNA to make a seamless transition between the fragments and make it all one beautiful piece of brand new DNA. So this is the process of DNA replication. Unwinds at the origin. It's going to split apart. We're going to lay down on the elongation phase, new DNA with DNA pole. And then in termination, we're going to come along and fix all those Okazaki fragments on the lagging strands, put them all back together, and that will be the end of this process with DNA ligase. So again, check out these videos to help reinforce this topic. It's a little tricky, but I think we can get it. So try the videos. I like lots of different ways to learn a process that is complicated. I want to hear it from different people because I may tell you one way, but maybe it clicks with you with the YouTube video instead. So definitely check out the videos to help with this process. Every time DNA replicates, at the end of the chromosome, there's going to be some extra base pairs there. It's kind of junk DNA, sort of, kind of. But it turns out those are important So these repeating strands of this DNA that doesn't code for anything, it's not going to be useful in coding to create things. It's just there for replication. Why are they there? So we have these repeating segments. Sometimes there's 100 of them. Sometimes there's 1,000 of them stuck on the end. And we finally figured out their value. It was actually Elizabeth Blackburn that kind of helped figure out their value. But there is, every time we are going to replicate, those snap off and those don't get recreated. One of them snaps off and the cell copies its DNA again. It goes through mitosis again, and then we snap it off. And then we go through again and again and again. So after, maybe 100 times, that cell can't copy itself anymore. It can't go through mitosis anymore because now it's going to start losing its good DNA and it's lost all of its junk. So there's an enzyme, telomerase, that comes along and sticks those pieces back on. It sticks the telomeres back on. So telomerase, the enzyme, is going to replace the telomeres. So here's our enzyme. Here's telomerase. It's going to replace the telomeres. And so this way, the cell can keep copying and copying and copying and copying itself. And it's not going to lose those pieces. It's not going to lose the telomeres at the end every time it copies itself because telomerase comes along and sticks them back on. So it's basically unlimited cell growth using telomerase. And that was discovered by Elizabeth Blackburn. She discovered this, what telomerase actually does. So we are going to see this happening in some stem cells use telomerase, but our normal regular cells can only copy themselves so many times before they run out of telomeres at the end. And those are the cells that you have. So telomerase will copy them and stick them back on so you can copy and copy and copy like crazy, and there's no end in sight. That is what cancer does. Cancer is uncontrolled cell growth, where it is copying like crazy itself. How do we learn about cancer cells and cancer cell growth? We learn about it through these cells called HeLa cells. Where did the HeLa cells come from? H-E-L-A. HeLa cells. And those HeLa cells came from a person called Henrietta Lacks. And so this is the story of Henrietta Lacks. It's a book. It's a really great book called The Immortal Life of Henrietta Lacks. If you're into reading books, if you are more the Netflix, Amazon Prime route, and you want to stream it, Oprah Winfrey made a movie about it too, about Henrietta Lacks. And so Henrietta Lacks was a Black woman in the 1950s, and she was married, had lots of kids, but she ended up getting HPV from her husband. He was unfaithful and brought back STDs, and so she was diagnosed with HPV. But yeah, especially now, we have vaccines out there to prevent HPV because HPV then turns into cancer, right? HPV has the potential to turn into cervical cancer. So that is what Henrietta Lacks had, but she didn't know what she had. And she went to the doctor and they took her cells and they pretty much sent her back. Didn't tell her that they took her cells, by the way. Didn't tell her that they were going to perform tests on her cells. They just, took her stuff and did whatever they wanted to. And this, again, was in the 1950s. So this is like my parents' generation. It's not mine, but it's my parents' generation. So it's not that long ago that this was happening. So the way medicine treated patients back then is a whole lot different than it is now. There were not the rules that we have now. They could really, it was crazy. You got to read a book. But she was treated, basically, in a nutshell, really terribly. She was treated terribly. Her family was treated terribly. They ended up taking her cancer cells and discovering that they grew. Those were the first time that cells were growing in a lab. So they took these cells, and the scientist took them and he sent them to all his friends. Look at these cells. They're copying themselves. They're growing in the lab. Check out these cells. So they use these cells to do all sorts of experiments because it's a ready-made source of cells. These cells are copying themselves. I can use these cells on all sorts of stuff. So lots of important scientific breakthroughs were using these HeLa cells. Where I want to say the polio vaccine was in thanks to the HeLa cells. But Henrietta Lacks didn't know that this was happening. Henrietta Lacks didn't get a dime for these cells. Her family didn't get a dime for these cells. They didn't get any recognition for the mistreatment that she had. So definitely, I love this book. Henrietta Lacks telling you about the mistreatment of people back in the day, back in the 1950s, and most especially the mistreatment of people of color back in the day. It is a great book. Highly recommend it. I haven't seen the movie, but I'm sure it's really good too. Check it out. Those are HeLa cells, and we literally still use them today. They are immortal. They are always copying themselves for cancer cells. And so it's pretty cool to find out the background of where we get some of those HeLa cells. It's come from Henrietta Lacks, HE for Henrietta and LA for Lacks are the HeLa cells. So cool story. Check out the movie, check out the book. It's really interesting about her struggle, her life and her family's struggle to achieve recognition and find out what happened with her and her treatment. All right, so last part in here are going to be our repairs. Before we get to those repairs, though, let's compare the DNA and the and prokaryotes and eukaryotes. When we compare DNA and prokaryotes and eukaryotes, we said that their DNA was located in the nucleus for eukaryotes and the nucleoid for prokaryotes. Eukaryotes have telomerase, which is going to stick on those telomeres so they never fall off, and we can just replace them with telomerase. We said that eukaryotes are going to have linear chromosomes, while prokaryotes have circular chromosomes. So these big long circles are all kind of coiled up on each other, but it's this big circular chromosome. It's not a line, it's a circle. Our origin of replication. We are going to have lots of them because we have lots of chromosomes and we're going to have lots of different origins. Prokaryotes have a single one. They're going to have this big giant circle with one origin right there, and then it's going to split off and it will replicate in a circle. So it's got one single origin on prokaryotes chromosome. Eukaryotes have multiple origins. How fast does DNA replicate prokaryote to eukaryote? Eukaryotes are about 500 to 550 to 100 nucleotides. And then check out the rate of replication for prokaryotes. It's 1,000 times. It's 1,000 times, 1,000 nucleotides per second when DNA replicates. That is crazy. But it also goes to show that prokaryotes can replicate super fast. They can undergo mitosis really fast. In perfect conditions in a lab, E. coli can copy itself in 20 minutes. 20 minutes. So that is due to this really fast rate of replication, and that their DNA is a little bit shorter than ours also. So they can replicate really, really fast. Again, they have a single origin, and that is the site where DNA replication is happening. They can rate, their rate is 10 times faster at least. And then they have circular chromosomes. Their DNA is present in an area called the nucleoid, and they don't have telomeres. We are going to have multiple sites of origin. Our rate of replication is a little bit slower. We have linear chromosomes. They are not circles. We do have a nucleus that's going to house our DNA inside of it. And we have telomerase, thanks to Elizabeth Blackburn's discovery, figuring out that the telomerase sticks on the telomeres, those little segments at the end that repeat. It sticks them back on so we can continue to copy and copy and copy and replicate. We said that mitosis had the cycles, the different segments. And there was interphase before mitosis, and interphase had G1, and then S, and then G2. And we said that there were checkpoints that the cell needed to check itself before it wrecked itself. It needs to fix anything that's wrong with it. So sometimes there's DNA, when it is replicating, has an error in it. We need to fix that. We need to proofread that. So some of those errors can be these guys here. So we might have a mismatch error. Here's our DNA it's proofreading, a mismatch error. So let's look at these guys. We have a G, and here's our G. What do G's match with? Are they G's and A's? Are they G's and T's? Are they G's and C's? G's need to be with C's. They don't need to be with this. That does not belong. That's not a proper match. That is a mismatch. So we need to come along with our DNA poll and see if we can figure out and correct the error. And now we have fixed it. G and C, we fixed that mismatched pair. So that works out really well. For that type of error, we can fix that. Sometimes, oh, excuse me. Sometimes we may need to cut out the error altogether. So here is our thymine dimers or T dimers. And in this one, this can happen. We all know to be careful in the sun, that UV radiation can cause changes to our DNA, can cause mutations, can cause skin cancer. Everyone's heard of skin cancer, hopefully. UV radiation can cause this condition right here where we have these two thymines, they're stuck together, and it is causing an error. So we may need to come along and break that up. So nucleotide excision is going to come along and bust up those dimers, and now they are just happy and healthy like they should be right here. So got rid of our thymine dimers. If DNA has these mutations and they're not fixed, there's mutations, and the mutations may result in not a big deal. Those mutations may completely change the protein that it's coding for. So depending on what's going on, sometimes these mutations are good, sometimes these mutations are terrible. It all depends. So we're going to get into what changes in the genetic code and for central dogma will play out later on. But these changes in DNA are mutations, and sometimes those are important for variation in the population. We said that sexual reproduction was really great because we get genetic variation. Mutations are another source of genetic variation. So sometimes if they're beneficial mutations, they're an adaptation and it helps something out. So different ways to think about that. Some of these errors we're able to fix. Hopefully we're able to fix them. And other mutations sneak on by and may cause drama later, or it may be a benefit later. We won't know until it happens. So that's DNA replication in this video. Next video, we'll go through the other steps involved with this process. If we're going to go through central dogma, we need to talk about transcription and translation in the next video.